So our next speaker for the Sciences Minisymposium is Dr. Robert Weinberg. He is the Daniel K. Ludwig Professor for Cancer Research and the Director of the Ludwig Cancer Center at MIT. He is an American Cancer Society Research Professor and a founding member of the Whitehead Institute for Biomedical Research. He was one of the scientists who helped discover the first human oncogene, RAS, which causes normal cells to form tumors. And his lab has also isolated the first known tumor suppressor gene, RB. He co-authored the landmark paper,
Perfect.
Hallmarks of Cancer. You want me to stop?
Continue.
Okay. He co-authored the Hallmarks of Cancer paper in 2000, and he also redid the paper for Hallmarks of Cancer, the Next Generation in 2011. It is the most cited paper on Cell, and he is extremely well decorated in his field, and I don't think he requires any further introduction. It is my great pleasure to introduce Dr. Robert Weinberg. Bob, if you would like to go ahead.
Thank you very much, Annabella. I'm most pleased and flattered to have been invited to give a talk today to this symposium. As I mentioned, Annabella, before, I'm going to talk my own career because she thought it would be illuminating for some people. Not that it's so different from any other people's careers, but maybe there's some interesting take-home lessons. I also told her that if you have questions during the middle of my talk, send them to Annabella and she will interrupt me with a question, perhaps when it's still fresh in people's minds. And as a consequence, Annabella, you can still see me? Yes?
Yes, sir. Yes, I can see you.
Okay. Okay. So as a consequence, Annabella said, how did I get into cancer research? Where has it taken me? And why am I doing it? Well, let me start at the end and say, to my mind, a successful career is the career of someone who actually enjoys doing what he or she is doing. Some people talk about somebody else as being a big success or being a big failure, but in the end, the big successes in life come from people who actually enjoy the everyday job of whatever their vocation happens to be. And this may or may not involve recognition. Annabella mentioned recognition I may have achieved, and I have on occasion in my career, but that really wasn't very meaningful for me compared with the fact that I enjoyed the everyday job of running my laboratory, of wrestling with scientific ideas, of writing them out, of keeping my brain working. That was the great pleasure. Asked me to talk a little bit in retrospect about how I got to doing what I'm doing. And in truth, I started out as a medical student or a pre-medical student, actually, because that's what boys did those days. I happened to have gone to MIT. Not that I knew anything about MIT, but because my parents had friends who sent their sons to MIT. And so more by luck than anything else, I ended up by happenstance at MIT. After I'd been at MIT for a semester, I heard that doctors needed to stay up all night to take care of their patients. And then and now, I needed my sleep. So I decided a career in medicine wasn't for me. I needed to do something which would allow me to get a good night's sleep. So I opted out of medicine. I'm telling you all the little stupid twists and turns, which have dictated my trajectory in life. In those days at MIT, the molecular biology revolution was just starting. The genetic code was being deciphered. And in 1973, I heard as a junior, a genetics course in which was described how DNA makes RNA, makes proteins. And then I realized, wow, the entire biosphere can be understood in terms of this single rule. It's not that biology is an infinitely complex phenomenon. Instead, there are a set of fixed rules, fundamental rules that govern all biological entities on the planet. And in fact, that turned out to be true. But that insight was so powerful for me that I decided from then on to become a biologist, indeed, a molecular biologist. Now, in truth, I was not very happy at MIT as an undergraduate. I was surrounded by engineers with whom I shared little in common, and I had a very mixed academic record. In fact, when I teach freshman biology at MIT these days, and I did do it for 20 years, in the first semester of in the first lecture of the semester, I say, class, I have a confession to make to you. When I took this course in 1961, the same course that I was then teaching almost 60 years later, when I took this course in 1961, I got a D, and they all start clapping excitedly, thinking it gave them permission, them license to get a D. But in any case, MIT graduate school, not because I had good grades. I didn't. In truth, I couldn't get into any other graduate school because of my written record. But at MIT, they took me on personal recognizance. They actually knew me. And so on that basis, they took me in graduate school. And there, for the first time, I really enjoyed myself because I was surrounded by biologists and no longer that much by engineers with whom I had almost nothing in common. And it was a very exciting and interesting time. And I started working on RNA, the messenger between the DNA and the proteins, more specifically on ribosomal RNA, which is the factory which allows proteins to get made. And so in those days, and now we're talking about the late 1960s, we already had electricity in the wheel then, believe it or not. In those days, genes had not yet been cloned. And so one didn't really know what messenger RNA really looked like. Mammalian messenger RNAs. One had done that with bacteria, but not with eukaryotic messenger RNAs. And so I decided that to study messenger RNAs from mammalian cells, the best way to do that would be to use a cloning experiment of nature. Because, for example, when SV40 virus, simian virus 40, infects a kidney cell, the progeny of that initial infection may number as many as 100, 200,000 copies of the parental viral genome that went into the cell and initiated the infection. And I said, wow, we can use that DNA, which the virus provides us, cloning itself, as a way of analyzing the messenger RNA. And so I started working on SV40, not because it happens to be a tumor virus, which it is, but because it represented a self-cloning vehicle for making copies of a gene. When I say a tumor virus, I refer to the fact that already by that time, it was clear that there was an array of different viruses floating around in the world. And some of them, when they infected animals, would actually create different kinds of cancer. I worked with SV40. It's noteworthy to say, not because it created tumors in mice and hamsters, but because its genome, it cloned its own genome. And that itself was quite extraordinary. In fact, I had an earlier encounter with SV40 because when I was in elementary school in the Pittsburgh Public Schools, Jonas Salk, who was in Pittsburgh, tried out his new poliovirus vaccine, a trial for all the public school students, the pupils in the Pittsburgh Public School system. And I was one of thousands of kids who were exposed to the poliovirus vaccine together with a healthy dose of SV40, which was contaminating the virus stock that was used to give myself and others protection against polio. The SV40 virus, which at that time was already known to cause cancer, albeit in rodents. Well, as it turned out, one could use SV40 DNA to study messenger RNA. And so, and I moved, I did 2 postdocs. And then I was called back to MIT because of another almost accident. I was working in a laboratory at the Salk Institute in La Jolla, California. The man running the laboratory was a man named Renato Del Becco, and Salvador Luria from the MIT biology department came by, and he walked by in the lab and he saw me there, and he came over to me and he said, "You're coming back to MIT." Mind you, he didn't ask me if I wanted to come back to MIT. He just told me that would be my future fate. And so what could I say? I didn't even have to look for a job. He told me I was coming back to MIT. That wasn't the worst thing in the world. My parents were on a rapid decline, and so I could move back from La Jolla, California to be at MIT. And so there I was, back at MIT after having left it 3 years earlier. And here I am now at MIT. I'm still at MIT now after some interruptions. I've just told you about them. 60 years and 1 month after my parents dropped me off at MIT in 1960. So you might say, in a sense, I'm a stick in the mud. I stay in one place and don't move around very much, and you wouldn't be wrong, but I have no regrets. There are some people in life who are academic gypsies. They move around from one job to another, one job for 5 years, another job for 10 years, another for 3 years, each time tearing up their roots, saying goodbye to all their friends and trying to make new ones. And I've always felt comfortable being in the same place and having and building a network of friends that has stood by me all these years. I've never regretted moving around because I've always enjoyed my surroundings. I'm not somebody who is instinctively against everything around me. You know the old story of a man who's shipwrecked on a desert island, and he climbs up onto the beach, and he looks around to the left and to the right and says, if there's a government here, I'm against it. That's the kind of attitude some people have who are always against wherever they are. And I've kind of been happy in that incarnation, being in that academic department in the MIT biology department. Now, I told you that I was working on SV40 tumor virus because it cloned its DNA prior to the time one could use bacterial vectors to clone DNA. And when I went back to MIT, I was working in the lab adjacent to David Baltimore. He had just isolated the enzyme reverse transcriptase, and he was working on a new class of viruses called retroviruses, which it turned out were causing leukemia in mice, and others cause leukemia in chickens. Now, in truth, neither David Baltimore nor I really cared that these viruses were causing malignancies, cancer in mice or chickens. We thought they were pretty interesting just by the way that they replicated their DNA or their RNA. And so my exposure to these retroviruses, which began in 1972, again, didn't come from any interest in cancer pathogenesis, in the development of cancer. It came from the fact that these were very interesting viruses to study in terms of their life cycles, how they could multiply in cells. You may know, by the way, that when David Baltimore and Howard Temin discovered the now notorious enzyme reverse transcriptase in 1970, and by the way, a big discovery for which Baltimore later received the Nobel Prize, when they discovered that enzyme, everybody thought that on its own justified Temin's theory that retroviruses were causing cancer in human beings. And as a consequence, in 1971, Richard Nixon signed the famous congressional act, the War on Cancer. The idea being that with the discovery of the enzyme reverse transcriptase and with the validation of retroviruses, one could now discover all of the viruses that were causing cancer in human populations. And so after the following 5 years, there was a mad scramble to find all of the retroviruses that were causing human cancer. And that mad scramble, with dozens and dozens of labs participating in it, fell flat on its face. It was a total failure because it turned out that retroviruses don't cause common human cancers. Very rare cancers, let's say in the Caribbean or in the southern islands of Japan, but the whole scheme was totally wrong. And the whole war on cancer was misconceived as being a way finally to discover the causes of human cancer by studying retroviruses. And by the way, because retrovirology was so intensively focused upon in the 1970s, when the AIDS virus was first confronted in the early 1980s, it took a matter of 2 or 3 years between the time that the syndrome of AIDS, acquired immunodeficiency syndrome, was described to the time the virus was isolated and characterized in molecular detail. An extraordinarily short period of time, a testimonial to the fact that the retrovirology, which had been studied in the previous decade for a totally unrelated reason, yielded a wealth of information that enabled one to study a very closely related virus, that HIV, that was actually responsible for applied immunodeficiency syndrome. So far, my interest at least had nothing to do with cancer pathogenesis. It just happened to be that I and people like me were studying tumor viruses, cancer viruses that went into organisms and instead of multiplying and killing the organisms, gave them cancer. In 1977, 78, I had an idea that maybe some of the techniques we were using to study these retroviruses could actually be used to answer an interesting question. And the interesting question was the following one. By that time, it was known that carcinogens cause cancer. It was also known through the work of Bruce Ames, then and now still at the University of California, Berkeley, that carcinogens are often mutagenic. By that I mean carcinogens often act by damaging DNA. And so the deduction from that was that to the extent that cancer cells are induced to become cancer cells through exposure to a carcinogen, they must be subjected to the mutagenic actions, to the mutation-inducing actions of some chemical carcinogen. And that in turn led to the further thought, maybe in the DNA of chemically transformed cells, these being cells that were converted to a cancerous state by exposure to a chemical carcinogen, in the DNA of those cells, there may actually exist mutated genes whose mutation was actually directly responsible for the cancerous state of those cells. And so in 1978, I got a new graduate student. And that new graduate student had been previously with another professor at MIT, and he had a big blow up with the other professor who threw him out of his lab. So when this new graduate student came to me, he couldn't afford to have another blow up with another professor at MIT. And he actually did what I asked him to do, which is quite unusual in my own experience. He undertook an experiment to see whether he could find DNA in chemically transformed cells that carried the information for cancer. And in fact, that experiment began to succeed. It succeeded very nicely. I had the idea of doing this kind of experiment during the blizzard of 1978 when Boston was blanketed in 3 feet of snow. And during the time I was crossing by foot the Longfellow Bridge in Boston, I had this idea of how to look for chemically transformed DNA that caused cancer. And I thought, gee, this was a terrific idea. I had the first original idea of my career. And so I was very pleased with myself for a matter of weeks until I discovered that exactly that idea had been published in the journal Nature some months previously by someone else. And clearly I must have been aware at some level or another of this other publication, somehow forgot how it appeared, and somehow thought that it was somebody recently arrived graduate student to start working on the idea. Now, at that time, in fact, the person who had posed an earlier idea was on the job market. And he or she, I won't even identify him, gave seminars at Cold Spring Harbor and at Harvard Medical School. And I went to one of the seminars given by this person, and it was extraordinary. It was extraordinary in the depth and the breadth of the data. There was evidence presented in this seminar that was more than my lab could possibly accumulate and generate in 10 years' time. Beautiful data. And so I was, having gone to that seminar by the man who had first published this idea, having gone to that seminar, I was both impressed and depressed. Impressed by the quality of the data, depressed by the fact that the work I had wanted to do was already done. He already had done all those experiments. And so for the first time, an original trajectory for me, an experimental trajectory, was effectively excluded because I was already an also-ran. The work had already been done. Still, we trudged onward. And actually, this person was actually invited to come to MIT to interview for a position, indeed, doing essentially the same thing that my lab had started to do. And this was a rather depressing piece of news because it meant that my superiors had little confidence in my ability to carry forward this very interesting work. A week before this person was to come and give a talk, Baltimore came and told, David Baltimore came and told me that that person who published the work had just been thrown out of his lab, thrown out of the lab of his postdoctoral mentor. And as it turned out, the idea that I was working on and the experiments that had been used to justify it were all dry labbed, as we said at the time. In other words, they were written out in a laboratory notebook without the actual experiments ever having been done. And so this was a rather awkward position for me to be in, working on an idea which, as interesting as it might have been, was soon thereafter under a cloud of suspicion, because the first demonstration, the first report of the idea in the literature had in fact been the subject of a major, we can call it fabrication. But still, we trudged on, and by 1979, published what ultimately was the most important paper in my career, which demonstrated that within the DNA of chemically transformed cells, such as human tumor cells, by the way, there actually is oncogenic information. By that, I mean to say cancer-causing information carried in the DNA molecule. And that itself was very exciting because it represented, at least in my way of thinking, a watershed in trying to understand the development of human cancer. So that's the way I've been yanked back and forth between various kinds of cancer research. It's been very interesting the whole time. I've met lots of interesting people, but I would say something else. There's lots of people who go into cancer research because they've lost a relative from this terrible disease. And in truth, these people are serious and well-motivated, but they aren't really well-motivated for the proper reason. Because the reason one should be doing cancer research, the real reason, the reason you can succeed in doing it is not because you're well-motivated because you lost a relative. That's not really the good reason. The real reason to do cancer research is that you actually enjoy doing it. If you enjoy doing it, any kind of research, you may actually excel. And if you're doing it just for some high and lofty moral reason, that already is hardly compelling in terms of preordaining what kind of what kind of career you will have. You need to enjoy what you're doing. And as I've tried to indicate to you, I landed on my feet in what I did, not because of any preordained plan, not because I planned to have a career in this or a career in that, but through a series of accidents. And I realized afterwards something I've long known, and that is I would have been just as happy in my life had I become a carpenter. I'm right now sitting in the cabin that my wife and I built in New Hampshire with our own hands. I kind of enjoy that. I kind of enjoy working with my hands as well as working with my brain and moving back and forth between working on my laptop, doing science and communicating via Zoom, as I am with you, and going out into the garden and building a rock wall or planting or doing weeding. That is to say, this oscillating back and forth between working with my hands and working with my mind. I'm not saying other people should have that kind of predilection, but I am saying that each of us is wired up differently. And to the extent that I enjoy my career, it has been extraordinarily enriching because, I say for the thousandth time, I've enjoyed it so very much. That's really what's made it so unusual for me. Not the recognition, again, not the recognition. It's been simply the day-to-day task of working, of teaching, of interacting with my students. They are what has made it so worthwhile and so exciting. Annabella, having said all that, might there be some questions, or should we postpone those questions until later?
Yes, I believe we will take questions at the very end. So if you have any questions for Bob, please put it in the Q&A button available in your doc. And thank you very much for your talk, Dr. Weinberg.